part 2. In this chapter, we're going to be discussing mostly gene expression and some other things that go on about the molecular biology of microbes, some of that. So we're going to get started with gene expression, which this is how we're going to go from what DNA says to polypeptide. How are we going to go from genotype to phenotype? Okay, gene expression is the process in which the information that's in our DNA, the information encoded in DNA, actually goes through to make an RNA, to make a DNA, and all that. Protein. So basically, we're going to go from DNA to RNA, RNA to protein. Okay. Now, it has 3 distinct steps: gene activation, transcription, and translation. Gene activation is going to be the part that happens in a gene where a gene is. There's an area called a promoter, which tells the gene, tells the cell's molecular machinery there's a gene there, and we need to read it. So each gene has a promoter on it. Now, what happens in bacteria, which is weird, bacteria are organized where there are many genes and there's one promoter and many genes. So please note in the bacterial genes, they may have multiple genes sharing one promoter in something called operons. But that's a little bit different way of regulating gene expression. Transcription, there's DNA to mRNA, and then translation is mRNA to a protein. Okay? So that we call that the central dogma of molecular biology, is that DNA is transcribed in a messenger RNA. Messenger RNA is translated into a protein. We actually just call that the central dogma of molecular biology. Now, genetic materials in a eukaryotic cell, it's in the nucleus, but in a prokaryote, it's not. It's not in the nucleus because there is no nucleus in a prokaryote. Alright, so one of the things I want to talk about is DNA with transcription. There's a few things where we make messenger RNA. It's a short-term copy. This is kind of like when you just write down a few bits of information that you need. You jot it down real quick just to use it, and you throw it away once you're done. Like a shopping list. Excuse me. A shopping list at the grocery store. You throw it away once you're done shopping. You didn't need that list for the long term. You only need it for that one shopping trip. That's what our messenger RNA is like. It's short term. Promoter sequences bind to the, they're found on DNA, and that's what's going to make an enzyme called RNA polymerase bind, and it's going to initiate transcription. Then you're going to have, it's going to go to 5' direction. There's a terminator sequence at the end that tells it to stop, and messenger RNA will be made during transcription. There are 3 phases or 3 stages. There's initiation, where it happens at a promoter; elongation, where we elongate it because RNA polymerase builds it; and termination, when it gets the terminator sequence of a gene, then it is terminated. Initiation, elongation, termination. Okay, so what I'm going to do is I'm going to go ahead and teach it transcription, and then we're going to go and go over how it happens. But I'm going to go ahead and teach it. This is how I like to do it, and it's my class. I'm going to do how I like. So, all right. So let's get started with some DNA. Let's say we have C, 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 G, 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 A, T, G, Gattaca, cat gag. Okay. Now this is a DNA strand, part of DNA. How do we know? It's got thymine in it, and this is called the coding strand. Okay, coding strand. Now we've got to find the strand that is complementary to it. Okay, and that complementary strand is based on DNA base pair rules, complementary rules, and this will be called a template strand. Okay, C binds a G, car in the garage. So G, 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 C, 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 A to T, T to A, G to C, A to T, T to A. Okay. Alright, and that makes our DNA sequence, our template strand, okay? This is our DNA molecule, okay? In a eukaryotic organism, this would be up in the nucleus. In a prokaryote, this is sitting there at the nucleoid region, but there is no nucleus, okay? Now, what we're getting ready to do is, with our DNA, is we're getting ready to make some messenger RNA, okay? Messenger RNA or mRNA. This process is called transcription. Transcribe it. Transcription. Sorry, I couldn't write it very easy to see there because of what kind of marker I have. Transcription. I can try to get it. Transcription. Okay. Transcription. About as small, not neat as I can get it with this marker. Okay. Now, what we're going to do is we're going to bind this template strand. Now, basically, I'm going to show you what happens. Okay. I'm going to show you kind of how it happens, and then what we're going to do is we're actually just going to draw it a different way. So, all right. So basically, we have our DNA. Okay. Our coding strand. And we have our DNA here, our template strand. What we have is a region here called a promoter, okay? It's called a promoter. An enzyme binds to this promoter called RNA polymerase. And what RNA polymerase does is polymerizes messenger RNA. So it will move down the template strand, okay? And what it will do is it will synthesize, as it does, a messenger RNA molecule, okay? And that is called transcription, okay? Now, don't draw this like in the middle here, like draw it somewhere else. I'm doing this quick sketch. You don't even have to draw this if you don't want to. This is just me talking about what we're doing, okay? Alright, now what we're going to do is we have G binds to C, 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 okay? G, 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 A, T, G. Oh, wait a minute. Wait a minute. That is RNA. It's AUG. This is RNA. Alright. Notice that this messenger RNA looks just like our coding strand here, except the T's became U's. Okay? Alright. Alright, now, we made our mRNA, we've done transcription. From this messenger RNA, what we use to do some work, if you had me for A&P or general biology, you'll know about it, is we need a translator, okay? We need a translator. We need what is called an anticodon, which is found on a molecule called tRNA, okay? Now, the anticodon, which we're going to find in just a moment, but that is transcription, and we're going to pick up with this drawing, and crap, it froze up. Let me see this. The app that I use to capture my drawing, it just froze up, and I can't draw anymore without uploading because if I close this app, it can't open with UJA running. You have to open it up first, so I can't draw anymore. So I'm going to have to upload this video and upload it in more pieces.